Hello, nice to see everybody again. With me, there's Dr. Chandrasekera, Charu Chandrasekera. Is that more or less correct? And I'm Marcin Krzykowski, and uh, we will be doing this interview with Charu about the NAMs that are being developed, but Charu is also involved very much in animal welfare and does her best to support, not only removal of animals from research, but also minimizing the suffering, which I believe is an extremely important um, part of the reason why we really do this. So, Charu, if you can introduce yourself, that would be really wonderful.
Thank you, Marcin. It's nice to be on your podcast. I think we met just a couple of months ago at a
Yeah,
3D cell
yeah,
culture conference in India.
yeah,
That
that
was
was
very
wonderful.
nice. How do I introduce myself? So, I am a Canadian scientist. I founded Canada's first and only center dedicated exclusively to alternatives to animal testing, the Canadian Center for Alternatives to Animal Methods, and its latest version called the Canadian Institute for Animal Free Science. So I have a diverse set of expertise. I was a former animal researcher, then I transitioned away to new approach methods, and I set up a new approach methods center here in Canada. And I work
Yeah,
with multi-sectoral stakeholders from government regulators to industry, academia, nonprofits. And my goal really is to educate the next generation to think outside the cage. I think that there's a lot we
that's
can
nice.
do with animal-free
Yes.
science, and it is really to bring all the stakeholders together and figure out what's wrong and how we can fix the problem. And this is not simply an animal welfare issue, but it is also a big scientific and a public health issue in many respects. So I try to address all of these different things, the technology aspect, the science policy aspects, the legislation, and a number of other things, which I'm sure we can get into.
Oh, yes, yes, very happy to. And one thing I want to highlight, like also from our perspective, we're at Real Research, we are building those research models. And this is also for the public to know that we think that animals are used for research because we don't have anything else. So you can do something in like a biochemical experiment, and then you basically, when you want to have organism-like research or results, you basically have to go to animals. And this is really the big promise of NAM's new approach methodologies, that we can replace the animals, and not only because that's suffering, but because it can be actually significantly better. So why make animals suffer and still produce very often irrelevant results? So what would be your story, since we had this little chat before the podcast, and I had also my experience with animal research, which was part of my motivation here. But what was your story when you did your part of your research with animals and then you decided to transition? How did that happen?
Wow, that was a long time ago now. When I
Yes.
when I think back, I think the first time I killed a rat was in graduate school. I had to
Yeah.
do a little rotation before I went to the lab where I did end up doing my PhD. I did not actually do much animal research at all, in fact, but I did this little rotation where we were doing, I honestly can't even remember the details, but I do remember that there were rats that were euthanized using dry ice. So there was
Yeah.
that
Oh,
big jar and there was dry ice at the bottom and you let the jar fill up with,
yeah. Yeah.
oh,
I know.
I don't want to even think about it, but it was the very first time that I had seen animals being killed. And here I was 22 years old, fresh
Yeah.
in grad school.
Yeah.
And I remember the professor putting like four rats into this one big jar and
Yeah.
they were gasping for breath. And I grew up as a Buddhist in Sri Lanka, and here I
Yeah.
am now Canadian in a Western animal research laboratory. It was shocking to me. I
Yeah.
mean, I was not vegan or anything,
Yeah. Yeah.
obviously.
Yeah.
I had used animals for other aspects of my life, but still to see an animal who was alive a minute ago now
Yeah.
being killed, and then they were gasping for breath. They were trying to climb up the side of the. So that was my very first experience. And obviously, I felt sad, and
Yeah.
it was also that Buddhist upbringing that I had, that you can't kill or that you don't kill. And I had never killed an animal before in my life, not
Yeah.
intentionally.
Yeah.
Maybe a mosquito or two, like growing up in Sri
Yeah.
Lanka.
Oh,
But
yes.
it was shocking. But then immediately I justified that I am doing this for the greater good. So that's how
Exactly.
sort of my animal research started. And I did not really do much. And during grad school, I think I used some tissue that was given to me from another lab that was killing animals, but I didn't actually really kill until I finished my PhD. And then I'm like, I need to become a well-rounded scientist and I need to do animal research because I had pretty much no
Yeah.
experience with animal research. So during my postdoctoral years, I went into animal research, cardiovascular research, heart
Yeah. Yeah.
failure, all that using
Oh, yeah.
mouse and rat models. And I think there were many, many, many different examples, but I think I saw mice, rats, rabbits, and dogs. And I
Yeah.
witnessed them firsthand. They were not necessarily all in my lab, but the lab next door
You've
and
seen
the
that.
next door
Yeah.
and all of that. You are exposed to that when you're in a research building like that. But I think the one moment that I can remember that really made me question, it wasn't just an ethical thing, though. There are two stories to this. There's an
Yeah.
ethical aspect of it, and then there's a scientific aspect of it.
Yeah.
So I think the ethics was, I remember I was looking at ischemia, reperfusion injury, and we had a number of transgenic animal models, knockout
Yeah.
models, and a number of other things. We were inducing heart failure in our laboratory.
Yeah.
And I remember one day I went down, I brought a mouse up
Yeah.
to the laboratory. It was a rat. We brought a rat up to the main
Yeah.
laboratory, and he was sitting there. And actually, come to think of it, there are two versions of this story because it sort of happened in one laboratory
Yeah.
and then it happened again in another laboratory in the same way. But one was a rat, one was a mouse. Either way, the story goes like this, where I brought him up and set him down on the lab bench, turned around to get the pentobarbital, to
Yeah.
do the euthanization. And that took a couple of minutes actually.
Yeah.
So he
Sure.
was sitting up there, and I took the syringe and I turned around, and all I needed to do was euthanize this animal and take the heart out. But I was just looking,
Yeah.
and the
Exactly.
animal was free. The little thing was sitting there and like pretty much cleaning his face, just like
Yeah.
my cat would clean his face, right? Like taking
Yeah.
one
Yeah.
paw at
Yeah.
a time, cleaning one side of the face. I know this is not a video podcast, but you can
I
kind of
know.
see, right? Like with one paw and clean the other side. And it was just very happy just doing that, completely oblivious to what was going to happen to him. And then he stopped for a second and looked at me. We made
Yeah.
eye contact for I don't know how long. I
Yeah.
do not honestly, looking back at it, I don't know how long it was, but we made eye contact. And I think in that
Okay.
moment, on that day, we had a conversation that I would never forget.
Yeah.
And I
Yeah.
think in him, in his eyes, in those eyes, I saw the exact same expression that I see in the eyes of my cat at home, a cat that I absolutely adored. And I've said this many times in many like interviews and other things. There was a cat who was in my life at the time who taught me all about animal sentience. It was
Okay.
her eyes. I saw in her eyes, her name was Mowgli. And I saw in her eyes this innocence and purity in animals. And she taught me that animals are just like us. They have the same capacity for joy, for
Yeah.
pain, for being mischievous and
Yeah.
playful, and so many different personalities in one. They're so much like us in that way. And I think I saw the exact same innocence and purity in my eyes. And in that moment, it just really clicked. What is the difference between my cat and
Yeah.
this rat in the lab?
Yeah.
I did not kill him.
Oh.
Um, that was the last time I ever even thought about killing an animal, but I did still stay in animal research, and I used animals that I got other people to kill and give me the tissue because I wasn't still thinking about leaving animal research at that time, but
Yeah.
I was looking at these animals not as simply a tool, a means to an end. I was looking at them
Yeah.
as beings who feel,
Yeah.
beings who
Exactly.
are conscious in many ways, right? And whatever the definition of consciousness is.
Right.
But then I still believed in the work that I did.
Yeah.
And the problem is that then the scientific issues were coming up. So the work that we were doing, we were looking at the role of adenosine receptors and interactions,
Yeah.
dimerization and all that with beta-adrenergic receptors and their role in ischemic revision injury. And, had all these different animals. We were doing all this work. And this is like 4 years into this research group. There were so many issues. Like nothing was reproducible between the different
Exactly.
laboratories. There were really big
Yeah.
laboratories doing this work. Nothing was reproducible. In fact, my project was to sort of settle one of these controversies, right? Because one lab
Yeah.
said this, another lab said this, and I was supposed to do something to show, which one was correct, or maybe
Yes.
come up with my own thing. It was a
Yes.
complete mess. Not
Yeah.
like everything that you can see with animal research that is not acceptable. I was witnessing it. So the 95% failure rate, all of these things. So, and at that same time, my father had a heart attack. And
Yeah.
then he had to have a quadruple bypass surgery. And I remember right after the surgery, I was sitting in this ward right next to his bed, and there were all these other people who had had surgeries, from like younger
Yeah.
people to older people, and I was just watching them in this ward in this cardiac unit in a hospital in Canada, and I think I forced myself to answer a question that I had been avoiding for a long time, and
Yeah.
that was, is the work that I'm doing with mice ever going to help patients like my father?
Exactly.
And the answer to me was extremely abundantly
No.
clear. It was a
Yeah.
no.
Yeah.
And I
Yeah.
think, so what happened was, just a few weeks later, I came back to the laboratory and I was talking to a professor who had been funded by the National Institutes of Health. I was doing
Yeah.
this work in the United States at the time. And, he'd been working on these mouse models of heart failure for well over 25 years. And I looked at him and I said, do you think these receptors are activated in my dad's heart during his heart attack? And he looked at me like, what the hell are you talking about? But I'm
Yeah.
like, do you think they were activated in my dad's heart? And he looked at me and he goes, how the hell would I know? We've never looked at this in the human heart.
Exactly.
That, I think that was it. That
Yeah.
was the moment
Yeah.
I decided I'm done.
Yeah.
So long story, but that's
Yeah.
sort of how I started.
Exactly. But I'm asking because that's part of our motivation. Like my story was like I actually had to finish my PhD, and that was with animal research. But once that was complete, I didn't write any other grant to do that. So we do have some grants, but we use animal models just as a reference. And then we don't use them for research. We basically try to avoid that as much as possible. And I'm talking literally about several animals per project. So that's nothing compared to hundreds or thousands used in other research. So and we do that basically to set a benchmark. So once we know what the benchmark is, we no longer have to do that with animals. So from this perspective, I think we have like your story is much more robust, much more elaborated, which is fantastic. In my case, it was really more of a one moment of like the same like yours, one rat, and I told you it was quite traumatic. After that, I actually had quite a few mice that I had to kill. But I think this is a lesson not only for us learning that the animals are suffering, but it's also that when you see what you just described, that this animal is I wouldn't say like us, but has this. You look at it with your emotions, you can really develop kind of a PTSD because of really having to go through this every day, basically, for many people. But what do you think has changed since then? I mean, especially about your question to that professor. So what can we do now, in your opinion, in terms of what are the other models that you think we can use? Because like we cannot tell to patients, well, we are not going to use animals, even with all the mishandling that we know exists, basically even happens every day. We have to have something better and tell them this is better because, and that has to be well documented. So what is your observation? You look at like we look mostly at efficacy, you look mostly at toxicology. So what is it that you see really changing for the last, I don't know, 10 years probably?
So animal use is quite varied, right? We use
Yeah,
animals mostly. If you look around the world, what we use close to 200 million animals, and then some countries
exactly.
don't count their animals. Even the United States, they don't
No,
count their mice and rats because they're not considered animals under their Animal Welfare Act.
no,
We'll talk about that later. But so when you're thinking about it, animal use is most prevalent in biomedical research.
yeah,
And then there's regulatory testing, right? Efficacy, toxicology, all of that
exactly.
for chemicals, cosmetics,
Yeah,
and drugs and therapeutics.
right.
And so when you look at the purpose of use is quite different, and the way you
Exactly.
apply animals to those disciplines is
Yeah.
quite different in each case. So I think when it comes to toxicology, I think the world is moving away from animal testing. It's quite obvious now, right? With
Right,
all of the changes that are happening with the US, with the FDA, with the
right,
EPA,
right.
and then with European Chemicals Agency or European Medicines Agency releasing these
Exactly.
statements and all of that. I think around the world we are seeing that transition quite
Yeah,
nicely. That with these new models, we are able to better predict human outcomes. And then obviously we can go into the different types of technologies, but
hmm,
there are still gaps in certain areas. But where I see the biggest issue is with biomedical research. And that's where
hmm,
most animals, if you look at animal use numbers, the European Union, United States, Canada, anywhere, the vast majority of them, like 40 to 50% at least, even
yeah,
more in some countries,
yeah,
these animals are used for biomedical research. And I think that that's an area where we can absolutely reduce
yeah,
the number of animals without these quote unquote safety concerns, right? So if
exactly.
you
Yeah,
really need to look at systemic toxicology and you don't have a way to do certain things without using animals at this point,
yeah,
let's just say, for argument's sake, that's justifiable
yeah,
because we don't quite
yeah,
have the ICH and the OECD guidelines yet. But what about if you're just simply taking a hypothesis that you concocted and
yeah,
you're like, oh, that's it. I'm just gonna use, 800 mice over the
exactly.
next 4 months, or
Exactly.
I'm gonna create this triple knockout mouse to do something. I think that's where we can really reduce the number of animals that we're using, and
Hmm,
that's what's not happening. So when it comes to talking about patients,
hmm,
how do you educate the public in that way, right? We are
yeah,
not losing something by switching, in my opinion. There are certain
hmm,
areas, like if you're creating another Alzheimer's mouse model, what the hell for? Right? It's
exactly.
been well over 100 years since Alzheimer described that disease in his human patients, and after a century of
Yeah,
extensive research, we only still have a handful of drugs, and they're not even
exactly.
right, effective unless you start giving them at a very early age. And most of the
Yeah,
clinical trials, right, 99.6% clinical trial failure
yeah,
rate for Alzheimer's drug development. So do you really need another mouse model? And I
yeah,
think these are the areas where we can deliberately reduce animal use, because in biomedical research, it's a lot easier to replace, in my opinion. If you want to look at gene expression in something, or if you
yeah,
want to like ask these basic fundamental questions at the molecular, cellular, and tissue organ level, they can easily be done with
yeah,
human biology based platforms first, right? Like, do
yeah,
you have to create a triple knockout mouse first? You don't.
yeah,
You can get a lot more information from human tissue
yeah,
and organ on chip and organoids or AI and so many other tools that we have at our disposal. So I think that there needs to be a way for us to transition away from animal use in biomedical research.
yeah,
And then I think for efficacy and toxicology, we're seeing the results now,
yeah,
results over the last 10 years, especially, that we've been exposed to a lot more systems that are now
yeah,
being validated for different contexts of use. And I think, I mean, there's obviously a lot of issues when it comes to them. I mean, we can
yeah.
talk about this for forever.
Yeah,
How do you validate these models? Do you do them against animal data, right? There's lots of different things, but I think that we need to intentionally
yeah,
make an effort to replace and reduce the use of animals in science, whatever that scientific purpose is, whether it's for safety,
yeah,
efficacy, or basic biomedical research.
yeah, exactly. So I think you would agree. There was this famous phrase that if you are a mouse, we can cure you from any cancer. You know, like you hear this on every second conference you go to. And the funny thing is, apparently it's true. So that's really funny.
Well, it is, and this is one of the things, right? In every time I teach, this is one of my first slides. I have it was a tweet that I actually saw. It was a
Yeah,
woman who tweeted in Canada several years ago, and she said, in the text or in this tweet, she said, my mother is having assisted death tomorrow.
oh
I'm
my
all
God.
alone in Canada.
Yeah,
And hashtag Alzheimer's. It was a very short tweet, but when I randomly came across that, this was not a person who was following me or anything, but when I randomly came across that,
yeah,
my heart sank.
yeah, exactly.
Because how do you tell? What do you tell someone like that? So every single time I do this, I put a copy of that tweet on a slide
Yeah,
and I ask my students, what, as a scientist, what do you tell that grieving daughter? Because a
yeah,
couple of days later, she tweeted and said, my mother passed
yeah,
peacefully. Thank you for all your wishes. A hashtag Alzheimer's. And to me, I wanted to respond to her and say that I'm so sorry for your loss. I'm so sorry that you had to go through this agony and that your mother had to go through this agony. Because if your mother had been a mouse, she would have had at her disposal, I don't know how many drugs to not
yeah,
even just treat, but to cure her condition.
exactly, exactly.
And that's the reality, right?
Yeah,
95% of drugs tested to be safe and effective in animals fail in human clinical trials. So we have to, at some point, come to that humble realization not every animal research study that we
yeah,
label, defend, and get approved and do as necessary is
yeah,
actually necessary.
yeah, sure. And it's really, I think there is I think science needs a little bit of better marketing because look, it seems so obvious that, well, if you cannot, obviously we cannot and we shouldn't use directly humans, especially on such a low level of research. Uh, and whenever there are people involved in the research, it's obviously very heavily regulated, which is fantastic, which was not true in 1950s, by the way. But what I'm trying to say is that on one hand, um, you can fool yourself by thinking this is an animal, this is a living thing, so there will be vasculature, there will be infiltration of the immune system, there will be stable temperature, there will be flow, there are so many things that seem to be like in humans. Therefore, from marketing perspective, I think it's very easy. We just got used to it, and it's like junk food. Like it it can taste well, but there is so much data telling you that it's killing you. It's not working. It's not giving you the thing. At the same time, there are still many people just following that way of living. And I think we do very similar thing in research. We do that kind of junk research as a whole. So I would say scientific part of the society that really it is, despite all the evidence, there is still, like you said, millions of animals being killed for research that is bringing very, very little. And I'm actually just preparing an episode that is describing penicillin. And it's really, I'm actually, the story, but it is really fascinating because if penicillin was supposed to be discovered today, it would not be discovered because it works in humans, but it doesn't work in many animals. And for some animals, it's actually extremely toxic. And that's really
Well, yes,
amazing.
didn't it kill Fleming's cat? I think Alexander Fleming's cat died
Yes,
after
true. And
something to that extent.
exactly. So that's what, the reason why I didn't release this episode yet, because I'm looking for good data for that. But the story about the cat is actually really interesting because there was a boy who had, I don't know which bacteria, sorry for that, but that's something I'm looking for details just to put that into the episode. And that boy had this infection, and doctors knew that he will die within probably like a week. That was the average lifespan for this disease. And normally the procedure would be like take a cat, inject the new medicine into the spinal cord because that was spinal infection. So, very, very, hard to treat. So inject the drug into the spinal cord of the cat and wait for 2 weeks. So what they did, because they knew the boy would die, they injected both the boy and the cat. The boy survived, the cat died. So you see, it's like on one hand, we are choosing the false positives because it's something works on animals, and we pursue that asset, we pursue this future hopeful drug. At the same time, there are so many false negatives. So there are things that are not working animals and could actually work in human trials.
So this is a very interesting thing because I mean, the literature is littered with examples like this. I mean, take aspirin. If you were to
Yeah,
ever put aspirin through the regulatory process today, it would never be approved for human consumption. And yet it's one of those things is like take an aspirin a day, keep the doctor away, right? It's like eating an
exactly,
apple for
exactly.
many people, taking
Yeah,
10 milligrams of aspirin a day or 100 milligrams. Um, it wouldn't, it's teratogenic in most of the
exactly,
lab animal species, but it's safe during all stages of human pregnancy. Look at, Vioxx, a
exactly.
simple anti-inflammatory that ended up causing stroke and failure, heart attacks, and all of these things. And you
Yeah,
look at all of these things, but they were tested, TGN 1412. It was
yeah,
tested in monkeys too, and yet we missed something very fundamental. And I think this is where we are missing the biggest point. I think that a lot of people have a tendency to dismiss
yeah,
alternative methods as simply they don't exist or they
yes,
don't work. What the hell does that mean? Like, seriously, I'm asking as a scientist, how can you simply say to an entire toolbox
yeah,
that it doesn't exist, first of all, or that it doesn't work? It works for what you want to use it for. And this is something that we really have to think about. So in your case, if you're looking for certain pathways, you're trying to
yeah,
identify a pathway and to see how to target a certain molecule, all of that, there's a lot that can be done with human biology based platforms to answer those
exactly.
questions.
Yeah,
And then you move on to the systemic interactions and things that cannot, let's say, perhaps be modeled fully yet
yeah,
without using an animal. So leave that as the last resort. And I think
yeah,
animals need to be the last resort, the very last resort,
sure,
not the first option. But right now, many people are just, I mean, otherwise, we should not be having 200 million animals per year for scientific purposes in this day
exactly.
and age. And
Yeah,
so that's the thing. Like, what do you do with this evidence? And I think it's really time for us to take the evidence into account. And just like you said, how would you discover penicillin today? And this is an exercise I do with my students,
exactly.
but with insulin, because
Okay.
everybody's always trying to invoke these insulin dogs, right?
Right,
Especially,
right.
one of the biggest contributions to science from Canada.
Yeah,
Yeah, sure. The dogs were involved. Please give them credit.
yeah,
And that's fine, but that was more than a century ago. And so
exactly,
you cannot say that yesterday's tools are appropriate for tomorrow's problems.
exactly.
And one of the assignments I get my students to do every year, and I've gotten very, very, very creative answers. And I'm like, if you were to
Yeah,
rediscover insulin today, how would you do it? And go through that whole process of getting it into humans. It can be
yeah,
done. And that's the
exactly.
thing. And I think that we need to be more cognizant of those
Yeah,
areas where we can absolutely replace and reduce and then save the unreplaceable. You cannot
yeah,
simply have these blanket statements to say that
exactly.
nothing is working.
Yeah. So I think I read that last year FDA for the first time banned IND filings that do not that contain only animal studies, which I think is a step to very good direction. I think this year they also banned grants. I think that was FDA and NIH that banned grants that involve animal studies only as well. So that is I think that's a very good, initiative. However, I do have a feeling when talking to mostly scientists, but I think both from academia and industry, that when you focus on let's say you're developing new PROTAC or new um, CAR T or something new, you're really specializing. You're extremely focused in that. So you have to be, let's say, in terms of CAR T, you have to know your target. You have to know how to make those CAR Ts in 50 different methods, whether it's per individual patient or you do it in a bulk for multiple patients. There are so many different questions you have to specialize in. And then there is this question of how do we test it? And I remember one conversation with one CEO, and she just told me, we just send it for animal testing. And that was so she was extremely smart. She was extremely clever. But for that approach, for when they designed it so well, they put so much time and effort into designing this therapy, they just throw it on something that exists. And I have a feeling like there's like the generation change that has to happen. So I'm very happy you're teaching your students what you're teaching, because they will be the future stars, hopefully. And probably they will be the ones who will finally replace and introduce those new methods at larger scales, because that's otherwise, we have this phrase, it's very hard to replant an old tree. I think, and I'm not, and don't take me wrong, I don't want to criticize that, because I learned a lot from very, very I would say
Old trees.
experienced scientists. However, there is this, once you really focus on one stuff, I have a feeling like it's very hard to have to take the other challenge on yourself. Okay, I will have to also be the best at, I don't know, let's say your cardio models.
I mean, this is a very systemic problem, right? It's a cultural issue. We have a scientific culture ingrained in animal
Exactly.
research, a culture
Exactly.
obsessed with curing disease in animals, a
Exactly.
culture that disregards human data as anecdotal, a culture that requires you to validate your human data using animal models, right? That is a
Yeah.
big problem. You've heard of the
Exactly.
animal methods bias. There are reviewers. I have personally experienced this. There was, I once submitted a grant to the Canadian Institutes of Health Research. It was to develop a 3D bioprinted lung model
Yeah.
for inhalation toxicology work.
Mm-hmm.
And the first time I submitted it, the reviews came back quite positive.
Mm-hmm.
And it was one of those, uh, small grants, right? You get funding for a year to do like a proof of concept kind of thing. It was one
Right.
of those things. So the reviews came back. They were only funding so many grants, and we missed it by like 3 or 4
Yeah. Yeah.
people. And so I'm like, oh, this is very positive. So we actually developed some preliminary data, submitted it to a bigger grant opportunity. Oh my God, you should have seen the reviews that came back. And one of the comments was,
Yeah.
you need a 5-year research grant cannot be justified without animal data. Are
Mm-hmm.
you kidding me?
Yeah.
Like, what is the point of doing that then? And I'm not the only person. I mean, this is why this whole animal methods bias thing started. There are a number of different groups who are really involved in bringing this to the limelight right now. And it happens to a lot of people. There's somebody that I know who's developing placenta on a chip, and they
Oh,
had to create a mouse model as well because the
really?
reviewers wanted data. Why? And that's the thing, right?
Yeah.
There is a tendency to dismiss human data. This is a big cultural problem. And I think that
Exactly.
with the next generation, we are going to see less and less of it. I'm
Yeah.
not blaming the people who are entrenched in
Oh
it,
no.
but
Of course.
because there's also other problems, right? You're living in this publish or perish culture,
Exactly.
and you really have to do certain things. You don't have money to venture
Yeah.
into other technologies, or you think you'll get behind, you think you can't get funded, you don't want to offend somebody, people don't talk freely. So I think there's a lot of
Exactly.
issues with our research
Yeah.
culture, whether, depends, it doesn't matter where you are in the world,
Yeah.
but there are certain issues. And I think as long as we hold on to that, that's
Yeah.
then it's really difficult to break through the barriers.
Exactly. And
I
yeah.
was going to say something I completely forgot.
Yes, please do.
No, what did you say? Okay, you're gonna have to edit this part out, but you said something and I had something really important to say. Um, I can't remember. Anyway, okay, that was
Anyway.
not the full answer I wanted to give. It was something else. I
So,
started
okay.
digressing.
Okay. I will try to edit this 49 minutes. Um, but anyway, so because there is this also this, I think, this notion that um, people need to start trusting those in vitro models. And as you know, the lab to lab variation is huge as well. Also, obviously in vitro models, one person, even like we have this very fresh experience. Like one of our customers actually used the same cells, the same models, and in their lab they grew slightly differently. And after literally going through step by step, we found out that one lab was using add DMEM and the other one was using DMEM 12. So this is a very minor shift, and still the results were different. They both look interesting, but morphologically very different. And I'm saying that because those very small details can actually make you, um, doubt what are you actually doing, because you can, you can have your SOPs, you can even have a GLP lab, at the same time a different batch of FBS if you use FBS, a different lot of your cell culture media. And all those small details, sometimes they really matter. Not always, but sometimes they do. And then it's extremely hard actually to compare the data. And I think this is also part of the problem, because on one hand, you can rely on extremely specialized CROs, but then they are very, very expensive. We know some of them. At the same time, you can try to do it yourself. But, I've spoken to so many people who said that they love those beautiful organoids publications, and they were never able to repeat, even half of those results. And that's really, there are labs who are extremely specialized, but I think the scalability is still very, very low. I'm just highlighting, another issue that could be really the reason why we see what we see.
So the reproducibility crisis is just the same for animal research as it is for alternatives. I think there's a lot that there are so many instances where one lab somewhere around the world would produce the same knockout mouse, and another one, around the corner or in the next country would do the exact same thing, but the phenotypes are completely different. And I have a big story about this. I think it was zinc T8. There's this whole thing about, like 6 different laboratories that created the mice at the same time. And then the same mice that were used, like the original colony was used in London and in Toronto, and the results were totally different because they were back crossed onto a different black mouse strain. So it's interesting that in that sense, we try to not ignore. There are a lot of people who try to deal with it, but it's not a problem that we have really been able to address. But when it comes to in vitro models, I think it's a lot easier to standardize. But the problem is there. Yes, many of these models are new, and we do need to standardize them, especially if you're going to use it for not just for exploratory research, but if you want to use it for validation studies for a particular purpose like toxicology, then you need to standardize them. And you need to come up with very specific SOPs with everything.
Exactly.
The growth media, all of that. Like from my experience, we've seen that. Obviously, the nutrient medium can alter not just the morphology, but all of the biochemical and functional properties of a system. But I think that's a lot easier to do. And the most encouraging thing right now is that there are so many different consortia, like the MPS, the Microphysiological System, the IQ-MPS consortium. They have a large number of guidelines for developing microphysiological systems, whether it's the lung or the liver or the kidney. And then the FDA just came up out with their own guidelines for that. And I think those are very helpful, so that when people are developing these new platforms, so you have to keep in mind the intra and interlaboratory variability and reproducibility. And at the same time, you can't dismiss them just because right? You can't just say they don't work. Like, this is what I'm saying. You can't put everything into that one umbrella. Maybe it works in one laboratory under certain conditions, and then you take it to another lab. And this is why you have these ring trials in some areas, and then you have these validation studies like the OECD, where you would have several member nations validating some of these technologies at the same time. And then try to come up with a standardized way of doing it. And we've seen this, right? When you look at the assays that have been developed to replace skin sensitization, the mouse and the guinea pig assays now have been replaced with essentially receptor activation assays, right? And you could it's been standardized, and it's a very
Exactly.
simple cell-based assay. So you can
Exactly.
kind of learn from those and then
Yeah.
use these new guidelines. Okay, if you have a liver model, what do you want it to be? What
Yeah.
absolute critical parameters do you need? What cell types? What
Exactly.
is this? And then perhaps you could grow them under different conditions in different laboratories.
Hmm.
But when it comes to incorporating these new methods into like ICH guidelines or OECD test guidelines, I think we are going to need to have to
Yeah.
standardize them.
Yes, of course.
And I just remembered what I was going to tell you earlier. So
Yeah.
the issue is that that lung model that
Hmm.
was rejected, the grant was
Yeah.
rejected.
Yeah.
They didn't, they didn't want to fund it without the animal data, even though I said that I will compare it to existing animal data. That wasn't good enough. I was not going
Of course.
to take mice and smoke them to death. But I ended up getting private funding from a
Okay.
philanthropic
Very good.
donor. We developed this lung model. It has 5 different cell types, 2 layers, an epithelial cell layer and an endothelial cell layer.
Yeah.
And we could, and I developed it specifically for repeated dose studies. And within
Hmm.
5 days, we can actually see fibrosis. We see
See.
beautiful
Yeah.
extracellular collagen deposition, and we sort of validated this model using an adverse outcome pathway for lung fibrosis that was developed by a scientist at Health Canada. And currently, Health Canada, they bought their own bioprinter and they are
Yeah. 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 Yeah.
independently recreating this model to validate it. And so here, you have these researchers at our, national funding agency saying this is a useless model and please do some testing with animals. And here's our regulator taking
Yeah.
our model
Yeah.
and
Yeah.
saying, oh, this is really cool because it is tied to an adverse outcome pathway. Let us like try to validate it and see how we can use this for inhalation toxicology. I think
Hmm.
there's a big disconnect. I think that in science, we think a lot about our own academic freedom, especially
Oh,
people in
yes.
academia.
Yeah.
I'm not criticizing the entire, academic culture, but there are a lot because it's important to have research. I'm not
Of course.
dismissing research.
Oh,
I'm
yes.
just
No.
saying that it just needs to be done far more collaboratively. Science is not there for you. It's not your grandmother's estate, right? You shouldn't be able to do whatever you want with public funds. It needs to be where you have a great idea
Yeah.
for trying to understand Alzheimer's disease, for example. Then like let's bring up like 10 other people in the same country who are really smart from different disciplines and we do it together. This whole doing everything on your own in these siloed
Yeah.
environments where you have so much competition and you just want to get your next publication out. And I think that's where
Yeah.
we're missing out on a lot of these things, that we don't have the freedom to
Yeah.
try a new model because you think you're going to lose out on 2 years of time, or you're not going to get a publication out, or that you're going to
Yeah.
be critiqued,
Yeah.
and you wouldn't be able to publish. So I think there are a lot of different issues to address here, but standardization would be important.
Of
But
course.
what's really important, what really ticks me off at times is that when people say they haven't been validated, they're not good enough. Please tell me who validated those, toxicology assays that were developed in the 50s and 60s? Who
Exactly.
validated them and what kind of standards? Like, I think the new approach methods are being held to extremely unreasonable standards at times.
Yeah.
I'm not saying they need to be sloppy. I'm
Of
saying
course.
that they're being held to unreasonable standards, and most of them are being asked to be validated against animal models or animal data.
Exactly.
But the approach that you use for new approach methods is very different.
In animal
When you
data.
are using, right, if you look at even skin sensitization, right, when you repeatedly apply a chemical on your skin and you develop an allergic reaction, how do you look at that in a mouse or guinea pig model versus how do you
Exactly.
capture that in an in
In
vitro
vitro
model?
model. Exactly.
It's a different type of thinking. This is what I mean.
Exactly.
When I'm teaching the next generation, I teach them to think differently. We do not have to recreate the same biological endpoints that you would be measuring in a mouse. We
Exactly.
need to think about human biology, how
Exactly.
you can break it down into bite-sized pieces, which is what I think
Exactly.
adverse outcome pathways are doing, that you can look at the molecular interactions, the cell, the signaling, the cellular, tissue, organism, population level, and you can integrate that information. And that's a very different way of looking at human biology
Of course.
than simply saying, let's just knock this out and see what happens. And so you
Exactly.
can't compare this type of information to a traditional animal model or traditional endpoints. So that
Yeah.
whole thinking, the standardization, the validation, the reproducibility, all of that has to change.
Yeah. And I think it's really changing, but slowly. And I think there is this very, very, very gradual understanding. And I recently also learned about the phrase context of use, which is the most important one in what you're talking about. So you basically define a context of use, like you said, fibrosis for smokers. And then how do you see, like you said, how do you define, how do you standardize the fibrosis in mice? So I will not give names, but in this industry where we are, there is one major competitor which basically didn't have to go through the same standardization as we have to do, but they are on the market for the last 40 years, which means that everybody knows, everybody has used that, and it's kind of accepted by diffusion, basically, like the animal models. So somebody started that in the 50s. At that time, there were basically no in vitro models. They were just barely starting with any 2D cell culture models. And now those animal models are really the standard, whereas you really can build much, much more sophisticated with much better definition of the context of use for the in vitro models, which I to be honest, I would doubt whether the animal models would fit the standards of the right context of use. So that's really something I think you can that in my opinion that will be the breaking point, where enough models will be so well defined that suddenly you will be able to scale them up and make it quickly, and suddenly you will see tens of those examples that it just works in vitro, and mouse or rat is just not doing that. So it will not make any sense. That's really my hope, honestly. And so what would be your take on really the advances? What is it that you see? Because I remember your presentation on your liver model, which was on one hand very simplistic. On the other hand, there was some very, very promising results that you showed. Also, you said like 7 types of different cells. By the way, is it already published?
No, it's not published yet. It would be, we are going through a little bit of, see, I'm not in this publish or perish culture right
Yeah,
now.
that's good.
Just kidding. But it's like, no, it hasn't been published. It will be soon. We are holding on for
Yeah,
a reason I cannot disclose at the moment, but
sure, sure.
we're hoping to get it out sometime soon. But yes, we have like 7 different cell types included in that.
So where do you think the whole, let's say, cardiotoxicity and liver toxicity, hepatotoxicity is moving? And how do you see those models actually being developed? And how do you see them? We all know that there are companies that really keep everything for themselves, and it looks like they're really well standardized internally. But when somebody drops a bomb on that lab, that method is gone. On the other hand, you know what I mean. Uh, on the other hand, there are those methods that seem to be, slowly, but being exported to other labs. They are very expensive, but it happens. Um, at the same time, the repeatability happens on the lower level, not on the top results that the first lab was really able to get. So what would be your take on the pace of advances, regardless of the regulation, regardless of the acceptance? How do you see the advances of the model research model, in vitro research model building to replace the animal studies? At the same time, I would even say accelerate them, accelerate the testing, therefore, getting rid of animal models.
It is a gradual process. So right now, I think the field is trying to address different questions, right? So what is missing, right? I mean, right now, the biggest thing that's missing is that everyone's complaining about is systemic toxicology, that you have
Yeah,
these individual models that represent individual organs. What do you
right.
do? We need the whole animal model.
Yeah,
And I think that's one of the big things that the field
yeah,
is trying to address, to create that human on a chip, especially for
yeah,
microphysiological
yeah,
systems, to have
yeah.
interconnected systems. So there's different technology frontiers happening, but at the same time, then it's really more about, I think my presentation in India was also about that. How do you take the existing models and put them into different contexts of use, or different
Exactly.
purposes, right? So if you have spheroids, for example, that are high throughput, you could use them in 384 well plates. What do you do with them? What are they best suited for? For screening, for doing like some preliminary tox studies or even preliminary efficacy studies. And then you move into something like 7 cell type 3D bioprinted tissue models, which is obviously low throughput, but higher physiological complexity. So
Hmm.
I think that you have your throughput versus your complexity and models that are appropriate for short-term acute studies versus long-term repeated dose studies, and then models that will give you quick information, right? If you just want to see to do some high content screening and look at some overall stuff versus something very specific like your model is only suitable for fibrosis, let's just say, well, then so
Exactly.
be it. Then use it
Exactly.
for that and then use another variation. So, our liver model, for example, we have different proportions of cell types for
Hmm.
steatosis versus cholestasis versus
Yeah.
fibrosis. So, you could
Exactly.
easily engineer them to suit your purpose. But I think what's really happening is that people are now taking these models and saying, okay, let's use it for this purpose. Let's use this one for this purpose. Where are the gaps? And let's build them now. So
Hmm.
one of the big examples, one of the things that's happening at the OECD at the moment is replacing developmental neurotoxicity. Now, this is,
Yeah.
as you can tell, it's an extremely complex
Oh
process. The
yes.
animal models suck, for the lack of a better word. There are no
Yeah.
gold standard mouse
Exactly.
model or rodent model that can give you that kind of reliable information on developmental neurotoxicity. But how do you also capture that? So it's not with one assay. Right now, they're developing what's known as a battery of assays that will
Exactly.
include like 12 to 18 different in vitro models combined together will give
Yeah.
you the answers that you want. You're looking at, myelin sheath, or you're looking at certain migration, or
Yeah.
so whatever, all the different cellular processes that may be involved, you're trying to capture it in a dish. And I think you need to factor all of these things into account. Your how fast can you do, so throughput, complexity, combining different assays, thinking about your biological endpoints differently. And what I am seeing that's really encouraging and exciting in the field is
Yeah.
that people are doing this. They are
That's great.
coming up with interesting ways to
Yeah.
combine these models
Yeah.
to capture different aspects of human biology.
Hmm. Yeah.
And especially with these guidance documents coming out
Yeah.
from different regulators around the world, we are going to be able to sort of say, okay, use organ on chip for these types of things, use, organoids for these, use AI for this,
Right.
use, physiologically based pharmacokinetic modeling, PBPK for this type of thing. And I think it's a combination of all of these different technologies, right? There
Yeah.
is no one model that fits all.
Yeah.
It's a matter of taking everything that we have and combine them as fit for purpose
Yeah.
to answer the biological questions that you're interested in.
Yeah. So I think what you're saying sounds to me like there are going to be new studies to educate people who will be this intermediate on those intermediate positions between the scientists and the decision makers who will basically know all the models. Because you see, I think you and I, we're both in this industry. And honestly, I know 5 toxicology models. I feel like I do know quite a lot of the efficacy models. I think I can, give you quite a good answer there. However, for toxicology, it's another cosmos. And probably liver toxicology, like the nephrotoxicology, all the other organs are separate, and all of them will require an extremely sophisticated expertise. I think AI will be very helpful there. So this is, well cataloging and, putting all of them together. However, I did have a feeling like from what, because I'm also learning from what you're talking about, and it's like I think the appeal of animal models, although I would say um, misleading, but it can give you a feeling that you have all of those models in one. That like this animal just contains all of those 500 different research models you would put in, and then if it's alive and it kills the tumor, this means that the drug works. I know it's misleading, but I'm just looking also for a way to understand the reason why it is so extremely, you know, it's so extremely hard to convince people who are not, you know what I'm talking about. Yeah, so that would be just my comment, and I think we would be, slowly wrapping up maybe. But if you have any, last comments, or your dream outcome for the next 5, 10, maybe 15 years, and you would like to share that, that would be really wonderful.
This is, that's going back to that culture issue. I
Yeah,
don't know why, as scientists, we are so reluctant. And by we,
yeah,
obviously, I'm not talking about you and I, but the culture. There are
yeah,
so many scientists out there who do not want to embrace change. And that's really difficult to understand because science is an evidence-based enterprise, not a belief system.
it should
And you
be.
would think that when the evidence tells you something that you would change. And I really don't have an answer to that. I'm not sure how easy it would be to change this culture, but it's really exciting to see the changes that are happening. I don't think it's happening fast enough.
Yeah,
I think that this is where you really need some top-down guidance. So when the U.S. Food and Drug Administration decides to, have
yeah,
updated legislation and come up with a roadmap and give guidance and say, we are doing this, that really forces the system to do this, right?
yeah,
And then, you have funding agencies like NIH saying, we don't want to accept
yeah,
animal-only grants anymore. That really
exactly.
go
That
like
is a
it
push.
trickles down. It's a big
Exactly.
push to do
Yeah,
that because you cannot expect individual researchers to really do that. They should be able to. And I mean, I'm not saying that there is no one. I'm just saying
yeah,
still the majority. It's really difficult to convince of the value of these models. And going back
mhm,
to your example, you can't like you could use it, but if the predictive value of your model is the
yeah,
as the reliability of a coin flip, what are you
exactly.
going to do? Like, are you happy
Yeah,
with a 43% carcinogenicity predicting model? It,
exactly.
it, you really have to stop and think. Animal models are not the be all end all. I
Yeah,
think where our thought process needs to go into this is animal models are just another tool, just one
exactly.
tool in that entire toolbox. And we cannot treat animal models as
Yeah,
the only tool in that box.
yeah,
And as soon as that mindset changes, I think it'll be a lot easier. But I
yeah,
think everyone has a role to play. And there's a
exactly.
big gap between um, the models that are being built, how to use them, and who is using them. So, industry,
Exactly.
regulators, policymakers, they aren't aware of all of it. Even when
Yeah.
we're in science, there are so many disciplines that we can't possibly know
Hmm.
what
Yeah.
every discipline's doing about what. So I'm not
Yeah.
an expert at computational modeling
Of
or
course.
AI.
Me neither.
So yes, when you think about it, I can talk all I want about microphysiological systems, but then this is why collaboration is so important. You need to have a
Exactly.
diverse interdisciplinary research team to incorporate these methods. But the easiest way to do it, I don't know what the easiest way is. It's
No, me
it's
neither.
that'll be the day when we figure it out. But I want to say that I'm hopeful. And I hope that over the next 10 years, we would see significant changes in different areas, especially when it comes to product safety testing, regulatory testing, that there'd be more and more um, new approach methods being incorporated into internationally harmonized test guidelines, whether it's the
Exactly.
OECD or ICH, ISO, all of these guidelines. I think that would be a big plus. But then at the same time, that's not where most of the animals are used. Like I said at the beginning
Exactly.
of the podcast, how do you bring these into the everyday average biomedical research laboratory?
Exactly.
And that's where we will see the biggest change. But 20 years from now, I hope we will have reduced our animal use by a measurable number, not 1, 2%, but
Yeah.
before I die, hopefully, I would like to see 90% reduction.
Yeah. Yeah. Me too. When you're talking, I actually thought about when you think about, back, let's move back 70 years to 1950, and all the scandals of actually doing experiments directly on humans. And I wonder if people those 70 years ago, if they had the same discussions, but and that there would be imagine a discussion where I'm trying to tell somebody we shouldn't use humans just for research, and they would say, "What are we going to do? How are we going to discover drugs?" You know, so I'm sure, like when we look at it right now, with all the ethics involved in clinical trials, we take it for granted. And I don't know, to be honest. I feel maybe I should get better educated, but from our discussion, because it's what we are talking about seems to be so obvious, and still there is a pushback. And maybe there was a similar pushback when people tried to be more conscious of not hurting other people as well. I'm just, I know I'm opening another Pandora box.
Yeah, that is for us to discuss another time when I am also more educated on this topic. But it's an interesting take on it.
Exactly.
Yeah, you do wonder what the mindset was, but I think it was
Exactly.
more of a, how would you even explain? I don't really want to get into the details of it, but when you think about all of those, you know, obviously completely unacceptable things that we did to
Exactly.
other humans, I don't know what the mindset was. I think it was a very primitive mindset of just like, let's see what happens kind of mindset. I don't think some of these
Yeah.
people who did these egregious human experiments ever thought to sit down and think,
Yeah.
how am I contributing to science? I think they were just, they
Exactly.
had power and they abused their power to answer some of their own intellectual
Yeah.
curiosity. But we are in a different state of mind, I think, when it comes
Yeah.
to,
Sure.
we've come a long way. But it wasn't that long ago when this happened, though,
Exactly.
if you think about it, right? We're
Exactly.
talking like 70 years ago.
Exactly.
So, or even closer than that in some cases. And we are really in that inflection point for animals too.
Yeah. I
And
hope so.
and uh, it feels like it because it's about time when you think about it. Like, what are we missing here? Why are we not doing our absolute best
Yeah.
to replace, reduce, and refine the use of animals in science? That the
Yeah.
3 R's principle came out so
Exactly.
long ago now. But we are not really doing it, at least in North America. I can tell you the
Yeah.
3 R's is an abstract concept. It's a box
Really?
ticking exercise. It's
Okay.
nothing more than a box ticking exercise. And I think I was telling you about these experiments, dog experiments
Oh,
in
yes. Yes. You told
Ontario.
me. Oh, yeah.
Right. So this was like last year, there was this big expose about heart attacks, induced heart attacks. So surgically induced heart attacks in
Yeah.
dogs, and some of them would last like 3 hours, three
Yeah.
hours.
Exactly.
They completely ligate a coronary artery and induce these massive 3 hour
Yeah.
heart attacks. And the chair of this animal care committee said on national radio, these are not your pet dogs who sit by your fireplace. And then the principal investigator for this project came out on national radio or TV a few months later and said, dogs don't suffer at all. These experiments, by definition, are classified as category D, which cause moderate to severe pain. And he was saying, well, they are under anesthesia. They don't feel the heart attack. But what about the rest of it? What about the thoracotomy surgery that you need to do for them to access the heart? Thoracotomy is one of the most painful surgeries
Yeah.
for a dog, and that's well documented in the veterinary literature. And there's recovery, there's pain after that when you lose 30 to 50% of your heart function and all of these things. So none of that counts. Only
Yeah.
like you're thinking they don't suffer. Yes, they don't feel the heart attack because they're anesthetized. So how do you get that thinking? To me, it's the
Yeah.
same kind of thinking that somebody thought when they were giving syphilis to
Yeah.
innocent men.
Yeah.
What were they thinking? And I could say the same thing about some of these researchers now. What were you thinking? And how could we be so disconnected that these animals are good enough to model
Yeah.
our biology, but they're not good enough to give the welfare and the standard of care that they deserve? If
Yeah.
they're so similar to human biology, then clearly their pain pathways must be similar too, if you go by that same argument. Well, then
Exactly.
why aren't we treating them better? How is it that yeah, you are
Yeah.
able to let these dogs suffer. And even like look at breeding facilities like Ridgeland, they were doing surgeries without anesthesia.
Yeah.
How do you allow that to happen in our culture of science? And please tell me, how many of those scientists are out there now protesting against that? I don't see any academics doing that. I mean, maybe there's the random one, but and so why aren't we speaking up as a community so we can better the system? So if the use of animals will be a standard for the next, let's say, 20, 30, 50 years, why aren't we at least consciously and doing our very best to transition it to a much better system? And I don't see that
Yeah.
happening. When I see it's a box ticking exercise, I'm not even exaggerating, because when I look at the justification for, in the animal use protocols, I read about 100 pages of animal use protocols from this heart attack lab. And, um, one of the questions is please explain how you are replacing animals in this project and refining and reducing, right? So they had to address each of the three R's. And for the replacement section, it was a simple answer. We need the animal model because we need
Yeah.
heart tissue.
Exactly.
I'm not kidding. This is how it's written. It's not a two-page, um,
Explanation.
defense of why they absolutely need this animal model. And quite frankly, most of what they did in these studies could have easily been done using human. They were doing some imaging, MRI and PET imaging, and there are other groups in Germany and the UK and Australia who've already published this using human trials or
Yeah.
human studies with informed consent.
Yeah.
And so some of these studies, and this is not the only one, there are so many. If you just go to PubMed and type in a random thing for a random disease or a pathway, you will see that there are a lot of animal studies that are absolutely unnecessary, studies where animals suffer a lot. So they are very high pain
Yeah.
categories of invasiveness. And then there are breeding conditions, there are living conditions, right? Mice, for example, are kept well below their critical thermoneutrality point. So they're constantly shivering,
Yeah.
freezing, their metabolism is high. That confounds data. Stress in
Definitely.
animals, biological stress is a
Yeah.
huge confounder. And so why aren't we dealing with all of these things? I don't know, when
Yeah.
you think about these things, good God, it's only like 2:17. I'm gonna need a whiskey.
Very good. So, Charu,
Yeah.
it's 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 a difficult topic, but at the same time, part of my intention was also just to highlight it. And I hope we're gonna get as much audience as we will be able to get. And as you know, some of our friends from Goa will be also on the podcast probably next month or 2 months from now. So I will be, talking with them on similar topics. Although, each and every one of them is in a slightly different corner of this industry. So it's really a pleasure talking with you. And we didn't talk about the way we met. It was your fight for the dogs in Ontario. So uh, maybe the next time with some more updates we can do that. But I just wanted to say that I think you're doing really great work, and that I think you told me also that you had some scientists actually working in those centers. Correct me if I'm wrong. I may be misleading that slightly, but they told you that they are not really protesting because they are afraid of their positions. So maybe the fact that you're speaking out is also helping them to kind of, maybe do something in the future as well. Because I really believe, like from the people I met in the animal facilities, most of them were actually would like to see change. And many of them stopped working with animals because of that. And so I think there is a huge support, but we need to, how to say that? We need to find a trigger to let everybody change it. And I don't know how it will be done, but I really hope that your initiative and many others like that will be the major part of the change.
I would really like to talk about the dogs and this culture of science when it comes to animal bioethics. I think we
Yeah,
should have a conversation about that at some point because what happened in Ontario is really worthwhile
yeah,
studying. It would be like an exemplary case study, actually. So very
hmm,
briefly, when there were whistleblowers who
right,
released all
right,
this information,
yeah,
and it made it to the front page of one of our two national. There's a right wing
yeah,
and a left wing newspaper. They're the two biggest ones. It made it on to one of the biggest platforms, and there
yeah,
was, outrage. People were
yeah,
outraged, 3 hour
yeah,
heart attacks on these animals and some of the videos, right? I don't think some of these animals had, enough pain control because some of the veterinarians that examined the undercover footage that we had, they were like, what is going on here?
yeah,
But so then, long story short, it got to the year of the premier. So, Premier of Ontario is like a governor in a US state. He's
yeah,
the head
yeah,
of the provincial legislation. And he said, nobody in our province is going to be experimenting on dogs and cats anymore.
yeah,
And 2 months later, he introduced brand new legislation
yeah.
to
And that's
ban
the change. That's the real
invasive.
change.
It's a real change, and there are lots of people who obviously criticize him. There were many, newspaper articles and podcasts and radio interviews, everyone saying he's interfering with, scientific freedom, blah, blah, blah. To me,
Yeah,
this is responsible leadership. This is what I was telling you.
yeah,
When and how is our system going to change? And I think that you need leadership like this and people like this to come in and say, that's it. You figure out another way to do things.
yeah,
And without that, like, because we are in that point where we
yeah,
see certain technologies coming out, we are this, and there's a lot of people who are still like, oh, I'm not going to change, or I can't
yeah,
change because I won't get my next grant funded, whatever the reason may
exactly.
be.
Yeah,
But then you have people like this, like our premier. I'm extremely supportive of this legislation. I think that
I
if
can
it
imagine.
passes, if it passes intact, the way it's written, it's history altering legislation in
That's great.
Canada and around the world. And I think it will start a very positive
Yeah,
feedback loop, positive change. And
great.
there were so many things that came out because of this, everyone defending. And one of the biggest things, maybe I think this could be a very good topic for us to talk about,
Yeah.
is freedom of expression in science. I
Oh, yes.
am literally one of like maybe a handful of people in Canada who are speaking out about this. A lot of people, I've had dozens of people reach out to me to have private conversations with me, but they don't want to do it in public because they're afraid of professional retribution. They don't want to be evaluated by their peers negatively
Yeah,
during the next grant review or next whatever. And they don't, and that's really unfortunate because we are scientists and we should be able to speak freely.
yeah,
And criticizing science is not
yeah,
criticizing what's not working in science is not unscientific. It is the only way science progresses, is if we
exactly.
figure out what's working, what's not working, if it's not working, why, and how can we
And correct it.
improve that. So when
Yeah,
it comes to things like this, I think more people need to speak up and be
of course.
open and have constructive dialogue.
Yeah,
I'm not, I'm not like constructive dialogue is very important to any system.
yeah,
And I also feel that, if would you ever get into a vehicle or an airplane that would fail 95% of the time? I know it's a dumb thing to say, but you wouldn't. And so why do you keep accepting some of
yeah,
these assays that have a 50% predictive value? It's the same thing. So why can't we talk about this? And why are you
yeah,
considered anti-science if you were to bring up problems in science? This is the only way we've
definitely.
ever resolved problems on this planet. So I really do wish, I'm just not affiliated with the traditional system right now, so I'm not afraid to speak, but I wish that there was more freedom so we could
Hmm.
all have these conversations because at some point in our history, preferably now, we need to change this.
Yeah, yeah, totally agree. So, Charu, I think that's a good topic for next podcast. Freedom of expression in science. So that would be a good one. We all know that. So, but that's going to be another an hour and a half. So for now, I really appreciate you finding the time and really thank you for being here, being honest, and thank you for your work. you're doing and for your excitement in this work, and hope to see you next time on another conference. I don't know how about you, but I do have some plans, but mostly industrial conferences for this year. But maybe an AACR next year or EACR. That's somewhere on my menu. So
Oh, thank you so much for
hope to
having
see
me. It
you.
was actually a pleasure meeting you, and I really enjoyed our conversations, this one, and obviously the many other conversations we've had.
Likewise.
And yes, and do plan to come to the 14th World Congress on Alternatives in Seoul
Okay.
next summer. So
Oh,
that's the
I didn't know
biggest
that.
conference series in this
Yeah.
field in new approach methods, the World Congress on Alternatives and Animal Use in the Life Sciences. But
Mm-hmm.
I'm sure, hopefully, I will run into you somewhere.
Yeah,
Otherwise,
yeah.
we
I'm sure we will.
communicate through LinkedIn and other means, I'm sure.
Exactly. Wonderful. So, Charu, pleasure speaking with you. Thanks a lot for finding the time, and I hope we will post it in a day or two. And yay! So,
Awesome.
thank you very much.
Thank you. Take care.